Hi, my name is Jenna Taffe, and I am from Drug Discovery Lab. And the title of our project is Investigating Antibacterial Effects of 4-nitrochalcone and trans-chalcone on Salmonella typhimurium. The goal of this project was to see if we could identify potential new antibiotics to treat typhoid fever, which is caused by Salmonella typhi bacteria and is a serious public health problem in the developing world, especially since Salmonella typhi, like many bacteria, are increasingly developing antibiotic resistance. And we used a different bacterium, Salmonella typhimurium, because it was safer in that it causes gastroenteritis as opposed to typhoid fever in humans, but does cause typhoid fever-like symptoms in mice, meaning that if our compound were to prove effective, it could be a good model to use in animals. Um, and we chose trans-chalcone as our compound because there had been some preliminary research in the literature that showed that it had some antibacterial effects, and because a previous group in this lab had tried to look into it. And we hypothesized that trans-chalcone would have antibiotic effects on Salmonella typhimurium, meaning it would either kill it or inhibit its growth, and that the effect would be greatest at the highest concentrations. And so what we did was we took trans-chalcone dissolved in DMSO, which is a solvent, because it would not dissolve in water. Um, and then we mixed that with salmonella dissolved in M9 media, which, unbeknownst to us at the time, was water-based. And we put this mixture into these wells, and then we would incubate them at 37 degrees Celsius, which is like body temperature, like an infection, overnight. And then we would measure using a spectrometer the absorbance of the wells, which could tell you how much bacterial growth there was. And we indeed saw a lot of absorbance interestingly, at the highest doses of trans-chalcone right here. And we thought at first that it was because it had some sort of nutrient that it was helping it grow. But actually, what we were observing was the fact that the M9 media, since that was water-based, the trans-chalcone was precipitating out. And it was clumping up, and that was what the spectrometer was detecting, was the actual trans-chalcone and not the bacterial growth. And so that was messing up our results. And then we also did not see a lot of bacterial growth in the 100% DMSO negative control because the DMSO was killing the bacteria. So we had to change to a different substance, 4-nitrochalcone, which uses this nitrile group here that allows it to have more polarity and more solubility in water. And we were able to dissolve it into a 50-50 mixture of DMSO and water, and that produced much better growth of the bacteria and Hart's negative control, and we did not have any sort of clumping issues in our results. We saw very similar results across each dose, and they were slightly smaller. We did determine that it was statistically significantly smaller, but just because something is statistically significant doesn't mean it's very large. In our case, it was only statistically significant because our sample size was so large. But yeah, you can see this is standard deviation is our error bars. The results are within one standard deviation, and the goal is to find something that's outside two standard deviations, like what we see here with the positive control, that's ampicillin. That's the kind of effect you want to see if you're giving someone an antibiotic. So while we may have found some sort of effect, potentially it's not a therapeutically useful one. And so we don't believe that nitrochalcone would be a good substance to continue to study, at least in Salmonella typhimurium. We do believe that perhaps other types of chalcones may prove useful if either they were to be modified like we did so that they're water soluble, or they could be tested perhaps using a different method that doesn't require water solubility. But we definitely did learn, and it's still useful even though as a finding, even though our results were a bit disappointing. And it's also useful that we learned that 50% DMSO is fine for Salmonella typhimurium, but 100% is not good. So basically, we're very happy that we had the chance to do this, and we would love to thank everyone who was involved in supporting us.